Today we will discuss about this ICAF and MyCAF. So let us first start with the what this ICAF and MyCAF stands for. This ICAF stands for Inflammatory Cancer Associated Fibroblast, and MyCAF stands for Cancer Associated Fibroblast. So these are two different kind of CAF or cancer associated fibroblasts that are found in the cancerous cells. The role of this ICAF or this we can say inflammatory cancer associated fibroblast is that it will cause local inflammation through the release of cytokines such as interleukin 6, as we can see here. And whereas the role of this myofibroblast cancer associated fibroblast is that it will release more extracellular extracellular matrix protein, as we can see in this graphical representation. And how these things are regulated through the signaling pathways. One of the that signaling pathways is this TGF-beta, and another signaling pathway is this JAK-STAT signaling pathway. And the competitions between these two pathways determine which one of this cancer associated fibroblast will out we can say will outreach others or will surpass others. And so because this TGF-beta upregulation of this TGF-beta signaling pathway will cause more of this myofibroblast cancer associated fibroblast, and whereas this upregulation of this JAK-STAT pathway will cause uh, more of this inflammatory cancer associated fibroblast. So and these kind of competitions carried out occurred during the tumor progression. So far in some of our previous videos we have discussed about this TGF-beta. Here we will consider how this TGF-beta interact with the different kind of immune cells. So this TGF-beta first we will start with this CD8 plus T cells. TGF-beta inhibit this CD8 plus T cells, or in another term it will prevent the activation of this CD8 plus T cells, and that leads to the decrease in the production of the cytokines by this CD8 plus T cells, such as this GZ, MB and PRF1, and that helps, and this cytokines helps in the immune response against the tumor cells. So that we can see here that this GZ, MB and PRF1 cytokines helps in the lysis of these tumor cells. However, this TGF-beta inhibit the or prevent the expression of this kind of cytokines through the suppression of this CD8 plus T cells activation. So this is one of the way by which these CD8 plus T cells TGF-beta inhibit this immune response. And another way that this TGF-beta inhibit immune response is via this CD4 plus T cells. What it does is it will upregulate the production of this FOXP3 in this CD4 plus T cells, and that will form this T regulatory T cells. And the function of this T regulatory T cells is to suppress the immune response, and thereby this will further we can say further reduce the efficiency of the immune surveillance, and that helps this tumor lysis cells to evade the immune response of the immune response by the host, innate immune response by the host. Other way, there could be other methods by which also this TGF-beta can play a role, and one of that we have discussed is in the previous slides, that is the formation of this extracellular matrix. So the extracellular matrix will provide a physical barrier to this tumor lysis cells, so that the immune cells will not be able to come and interact with the antigen that are found on the tumor lysis, unique antigens that are found on the tumor lysis cells, thus will not be able to trigger immune response. So this is another way by which this TGF-beta helps in the immune evasion of these tumor cells. If you need to understand more about this TGF-beta and cancer-associated fibroblast, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.